Hey folks, and welcome to another edition of Zog Science. Today we're going to be talking about how the cell cycle is regulated and what happens when that regulation does not go so well. So, why are we want to regulate? Well, different cells divide at different times, or sometimes not at all. For example, neurons don't really divide at all. Excuse me. We want to conserve energy use. We don't want cells just constantly dividing because that burns up a lot of energy. And as we all know, energy is really important. And we want to divide at the right times. You know, you don't need your skin cells to be dividing like crazy if you don't have an injury. But as soon as you get like a cut or something like that, you want them to start dividing a lot to be able to help repair that injury, right? Some experiments were done to kind of figure out whether or not there was regulation of a cell cycle. And so what they would do is that they would take cells that were in different phases from each other. They would fuse them together. And when they did that, the other cell would immediately move into that phase. So, for example, G1 and S phase cells would be fused together and they would immediately go to the S phase, or a mitotic cell and a G1 cell would be fused together and it would immediately go into the mitosis phase. So there's definitely, experimental evidence to support this regulation and why the regulation occurs. Okay. Now, in terms of how the system works, the system is kind of used a lot of times as an analogy would be a washer dryer system, right? So you turn the dial on the washer and you hit start and it kind of goes and it will stop at certain points. That's very similar to how this works. So the cycle is going to continue until it gets to some checkpoints. And at those checkpoints, there are some molecules that must be present in order for the process to move forward. So we're going to talk about kind of three main checkpoints. That's the G1 checkpoint, the G2 checkpoint, and then the M checkpoint. And we will get into kind of some of the specific features of the checkpoints. There are other checkpoints along the way, but we're not going to discuss those. So the G1 checkpoint is a restriction point in mammalian cells. It is really crucial. And what happens is that there are some signals involved. And if those signals are not present, then the cells will go into a non-dividing G0 phase. And most of the cells in our body are in this non-dividing G0 phase. Maybe because you're still growing, you might have more cells that are going through the cycle. But for me, at least as an adult, most of my cells are going to be in the G0 phase. They're not going to be dividing. They're just kind of, holding steady, waiting for the proper signals to come in place that are going to allow them to move forward. Some of the proteins involved are kinases and cyclins. And what you can see right here is this graph is showing us the percentage of dividing cells. And when the kinase activity is higher, the percentage of dividing cells is higher. And that's kind of one of the ways that they have figured out that they are related to one another. The G2 checkpoint regulates whether or not a cell enters mitosis, and it makes sure it's the cells check to make sure that the DNA is not damaged. And this is really important because when DNA is damaged, that can lead to things like cancer and other diseases. So we want to make sure that the DNA is put in place properly. It's not messed up. The way that this is regulated is that we have the CDK protein, which exists in the cell. CDK proteins, throughout the G1 and S and G2 phases, slowly build up over time. The CDK stays the same. But as we go through G1, S, and G2 phases, cyclin, this protein, builds up as long as the DNA is okay. If the DNA is fine, the cyclin will bind to the CDK and produce MPF, which is maturation promoting factor, or mitosis promoting factor is another way to think about it. And what that does is that if the MPF is present, if there's high levels of MPF, then mitosis will move forward. We will go into the mitotic stage of the cell cycle. Finally, we've got the M checkpoint. This basically makes sure that all of the chromosomes are lined up right along the metaphase plate. So basically, there is a sensor molecule that detects when the kinetochores are in the proper positions. And when they are all in the proper positions, that causes a signal cascade that leads to anaphase being initiated. So it's between metaphase and anaphase. And if there is a defective checkpoint, what that can lead to is what we call a segregation error, and you can end up with more chromosomes in one of the daughter cells than with the other daughter cell. And you don't necessarily need to know the specific molecules involved in this, but again, just kind of the overall what's going on, what is the usefulness of this checkpoint. We can also have some other external factors. So those are all internal factors. These are some external factors. A good example of those are growth factors. These are proteins released by cells in one part of the body that will stimulate other cells, or it could be a neighbor, but just stimulated or released by one cell that stimulates another cell. An example would be platelet-derived growth factor. So cells are going to be more likely to proliferate, and specifically fibroblasts, which are a type of connective tissue that kind of help hold things together. And it allows cells to pass the G1 checkpoint. So in this case, the growth factors enable the G1 checkpoint. Depends on which type of cell we're talking about, what growth factors do. This is really important because fibroblasts help promote healing of wounds. Again, they're connective tissue that help hold things together. So PDGF is released a lot at wound sites in order to help maintain and promote healing and promote your tissues to kind of hold together. We can also have inhibitors to the cell cycle. So there are kind of two different main ones. The first one is anchorage dependence. Animal cells must be attached to something in order to divide. If you just have animal cells that are floating around in a suspension in a liquid, they will not divide. Now bacterial cells will, but animal cells will not. So they have to be attached to something. So what they did was that they took kind of a petri dish and they put some cells in there. The petri dish, the cells began to divide. And they divided until they formed a single layer over the petri dish, and then they stopped. And we call this density-dependent inhibition. So as the density of the cells increased, it inhibited the other cells from dividing. And what plays a large role in this is the ECM, or the extracellular matrix, and the proteins involved. When they bind to one another, they can sense the fact that, hey, there are more cells around me, so I'm going to stop dividing. Okay, and so this is kind of what happens. Now, cancer cells, they will not care. They will continue to grow despite these density inhibition factors, and you can kind of see the structural difference between these cells, right? They look different, and cancer is mitosis gone wrong. So there are lots of different types of cancer. Some make their own growth factors. Others ignore inhibition signals. So there are kind of different classes of them, and each cancer is different. Even like, breast cancer has different ways that breast cancer can occur. So there's just a lot of different things involved in cancer. And that's one of the reasons why it's very difficult to develop cures for cancer, quote unquote cures, because it's such a varied and disease that it's very difficult to kind of figure that out. So abnormal behavior in the cell begins with what is known as transformation, where a single cell becomes cancerous. And because it starts off as a single cell, but mitosis has gone wrong, it begins to divide rampantly. Normal bodies destroy cancer cells. So your immune system is built to destroy cancer cells, but sometimes they evade detection, and they can become either benign or malignant. Benign is less serious. It doesn't spread throughout the body. Now, if you have a benign brain tumor, still can be a problem because it's going to be pushing on your brain and other things like that. But it's not as serious. Now, the more serious type is called malignant. This is where the cancer can spread throughout the body. It is a term would be an aggressive cancer, just because it moves throughout the body. And when that happens, that is known as metastasis, or you would say the cancer has metastasized. That's the spread of cancer cells to locations distant from their origin. So, for example, someone who has prostate cancer, it starts off in their prostate, but then it might travel through the circulatory system to the liver or to the heart or something like that. So kind of how that would work is we might start with something like breast cancer, where you've got a tumor that develops, and then eventually that tumor grows. The tumor grows. Go back. There we go. Tumor grows and it begins to invade some neighboring tissues. Eventually, it gets into either the lymph vessels or into your blood vessels and moves around and maybe goes into the stomach area or something like that. So the genetic basis of cancer, there are definitely some genes that control cancer that are associated with cancer. The two types we're going to talk about are proto-oncogenes and tumor suppressor genes. And in either of these cases, either a protein is being overexpressed, which means the cell cycle is overstimulated, or a protein is absent, so the cell cycle is not inhibited. So proto-oncogenes, they stimulate cell division, and the mutated versions are always on. So what that means is that the proteins that are being produced by these genes stimulate cell division, and because they're constantly being produced, cell division constantly keeps happening. So in this example, we've got a growth factor that activates a receptor, which activates a phosphorylation cascade, which maybe activates a G protein, and then we've got a hyperactive RAS protein, which is a product of an oncogene, issues signals, which leads to the production or the cell cycle being continuously pushed forward. We also have tumor suppressor genes. These inhibit cell division. The mutated version is always off. So in this case, the tumor suppressor genes have been turned off. Therefore, they are unable to suppress or inhibit cell division. Therefore, cell division continues to occur. So again, here's another example. Maybe we've got some UV light that damages the DNA that codes for a protein that normally inhibits cell division. So without that protein to inhibit cell division is allowed to keep moving forward. One of the kind of famous proteins is p53, and that is definitely something that you should be aware of as one of the kind of the classic inhibitor proteins that has been disabled by cancer. All right, that's all I've got for you today. Thanks for watching, and I'll see you next time.